Structure of chromosome. Good morning. Okay, so what we were discussing, you all aware? Exams are over. Your exams are over. Yes. Which school is this? Seventh day, no? Yeah, these 2 lectures or 1 lecture? 2, I suppose. Shall black marker there? Oh, इसमें मैंने ये क्या black? मैंने ये. So we were talking about absorption by roots, and what we have finished discussing was we had already finished the first part of the chapter. The first part of the chapter where we had already finished of what we had already been discussing about inhibition. What else? We had understood the concept of diffusion. We were talking about experiments on osmosis. First of all, we'll start with one question and then proceed with the chapter. Take one question that we will solve, and that is based on this concept. This was the last concept that we were discussing the other day. Osmosis. Okay? Osmosis. So this is just a recap of what we had studied the other day. Let us try to solve these questions. I'm going to write this down, answer the questions in one word sentences, one word, whatever is necessary. quite a number of such type of questions the other day we have done. Let's see how much you remember the concept. Try one more. Let's see. The different application of the concepts is very, very important in this chapter. How are you able to apply the concept in different situations, in different examples, in different questions? This type of question with this simple diagram, many questions you get. Don't take more than 5-7 minutes. Karo. Is this chapter over in your school? 7th day? Okay, so at least you know that. बैटरी खत्म है इसका?
Yes.
हाँ? अरे और एक ही लौटा लेके आए हो?
You want to talk about back sleeping.
अच्छा पूरा बैग उठा के लेके आए हो?
Yes, it's sleeping.
सारे के सारे एक ही बचा है. Finished? Anybody has finished? Did you finish? No. It's I'm going to teach you. the level of solution water in the pipe. ये कब होगा? That is what I wanted to know from you. कि कैसे तुम ये situation rise कर सकते हो? How can you reach up to a situation like this when the movement of the water will, the net movement of the water will be more or less zero? When will the situation come? The last day I explained it. When on either side of the semi-permeable membrane, the same stability is there. So how can you show that? LIN. Think about that number D. That is okay, but how now in this experiment what we will have to do? In this experiment, what should we do to bring a situation something like that? In this experiment, what will you do? This is the principle. This is the formula. This is the principle. How can this be achieved in this experiment? Think once. Done. Side. Done.
Should I have multiples? Should I have multiples for D2?
I don't really know. No, you are writing down the general theory. General theory मुझे नहीं चाहिए. I want to know what's changed. For general theory principle, that is okay. That is a standard concept that all of us we know. This experiment, what will you do to bring about that concept? Description नहीं चाहिए. You don't have to write long descriptions. Just in one sentence you have to write. Answers to these type of questions should not exceed than a sentence. The way he has written. I'll just show them. Just one sentence लिखना है. One word, one sentence. These type of questions, especially for Icse, you don't have to write any description for any question also on the paper. Unless and until you get those 5-mark questions. जहाँ पे they would ask you to describe something, explain something. यहाँ पे this is not the pattern of questions. Repeatedly, the other also 4-5 questions we have done. And I'm constantly trying to make you understand description नहीं चाहिए. One sentence में answer. And that will be, you'll be able to write down one sentence answer only when you are very clear about the concept. Description नहीं लिखना है. I'll tell you the core truth quite a number of times when you write एक mark के लिए paragraph लिख रहे हो ना, teachers will just cut that around. We generally just cancel it around. इतना description हमें नहीं चाहिए. Just one word, one sentence. Then only we are very clear that yes, the student has understood the concept. When you don't know the concept, तुम describe करने लग जाते हो. So वो नहीं चाहिए. Where's the attendant sheet? No, that's not the one. अच्छा.
आज मैंने नाम किया है.
Okay. All of you will answer. I think there is a problem with this question. No, most of you are not able to answer this question. Rest, you could answer. क्या लिखा तुमने बताओ? Number 1.
Semi-permeable.
Semi-permeable.
It does not.
कहाँ गए थे तुम? 2 हफ्ते बाद आके छींक रहे हो यहाँ पे. Now, write one feature of the Parkman's membrane. One feature of the Parkman's membrane. What is it? Semi-permeable. Okay, so this answer you know. What would you observe after a experimental period of time?
The water will be more salty.
Arey, you were confused the other day also. Today also you were confused. एक बार अच्छी तरह से देखो. कहाँ पे concentration ज़्यादा है, कहाँ पे which is a dilute solution, which is a concentrated medium. कौन सा dilute medium है बताओ मुझे.
ठीक है ठीक है.
ऐसा नहीं है कि ये आंसर नहीं तो वो आंसर. Not that. You should be very clear what you are saying. Dilute. What did I explain you the other day? Dilute मतलब जहाँ पे water ज़्यादा. Concentrated मतलब जहाँ पे water कम. Very simple. तो ये दो medium में which is the dilute medium? Weaker. This is the constant. अगर मैं यहाँ पे 1% भी लिखूं फिर भी तो ये concentrated है. आया समझ में? So I wrote that this is 10%. No? I wrote that this is 10%. So yes, this is obviously the concentrated medium. What would you observe after 6 to 8 hours?
After 6 to 8 hours, the water, the salt solution will increase.
The level of the solution in the funnel increases. So now this is the
And the
final.
water level decreases in the beaker.
And the level of water, if this is the initial level, it drops. I'm just telling you I'm just showing you this difference. I told you the other day also. This difference only to make you understand, but in 8 hours we will not see this much of difference. वो difference थोड़ा ही होता है. The process is slow. But this big difference I'm showing you, you will see in the figures only to make the concept clear. कि difference होता है, but it's not a big difference like this. अच्छा, give one word to describe the process. One word क्या लिखेंगे? What which process is happening here? We take this funnel. What is this funnel resembling? What is this funnel resembling in the real situation? What is this funnel resembling? A root hair cell or a cell. The funnel is actually resembling a cell. So cell के अंदर water is moving or water is moving out of the cell. What is happening in this process? The water is moving inside the cell. If I take this as a cell, the water is moving inside the cell or water is moving away from the cell? Inside the cell. So inward movement को क्या बोलेंगे? Endoosmosis. Give one word. Answer is endoosmosis. But मुझे ये movement of water नहीं चाहिए. I want to stop the movement of water. So what should I do? What are the different possibilities that I can do? What are the different changes in the experiment that I can do to stop this endoosmosis? What should I do here?
Can change the temperature to better.
Can change
The temperature.
Parchment paper. Can change the parchment paper नहीं. अगर parchment paper में change करूँ, then also there will be a movement of water either this way or this way. अच्छा. Yes, I can also use a particular type of something like the rubber sheet, which will not allow the movement. But no, I'm not actually talking about movement that way, but I'm talking about a movement जो stop हो, at the same time balanced भी हो. Yes, that way also it is correct. If I talk about the usage of a rubber sheet, movement may not be there, but will be huge concentration difference there. So let us take it this way. How can the movement be stopped at the same time, more or less a balanced condition on either side of the membrane?
By placing it in a hypertonic solution.
कौन सा? This one?
Outside.
यहाँ पे hypertonic बनाना है. ये water को substitute करना है by hypertonic. That is what you are saying, no? ये क्या बोल रहे हो तुम? What are you saying? ये जो movement है, मुझे इस movement को stop करना है. But I am not talking about changing the parchment. I did not write here by changing the membrane or something this way. I don't want to change the membrane, but फिर भी मुझे कुछ और change करके I want to stop the endoosmosis. I want to stop that process. Okay, never mind. Yes, the girl there in the corner. Bolo, what to do? What can we do? Whatever. See. और क्या change हम कर सकते? I don't want to change this paper. I don't want to change this membrane. I did not mention in the paper, in the question. ये नहीं change करना है मुझे, but still I want to stop by doing some other change. I want to stop the movement of water inwards. क्या कर सकते हैं? दो possibility, one or two of you have written one possibility, एक और possibility भी है. So what are those changes that we can do? He has written a correct answer. बोलो. Soham, no?
Yes.
हाँ.
Make the water in the beaker into a 10% salt
Yeah.
solution.
Either हम इसको substitute कर दे same type of solution से. तो either side of the membrane balanced हो गया ना? Will there be a net movement? नहीं. या तो एक और भी हो सकता है, या तो ये 10% solution नहीं लेके यहाँ पे भी हम water ले. मतलब whatever I take here, the same type of medium should be here. या तो in both the medium you take same 10% solution, or else in both the medium you take water. Why? How will the medium? How will the movement stop now? The movement will obviously stop because on either side of the semi-permeable membrane there's a balance state. Why does osmosis happen? To bring about a balance between the two medium. एक में solute ज़्यादा रहता है, दूसरे में solute कम रहता है. That's the reason the water is moving from a region of high concentration to a region of low concentration. है ना? So this is what you will write. So what should you write? How can the movement of the water be stopped? The movement of the water can be stopped. Please write down this answer and these concepts याद रखो. This type of questions are there in the paper and you should easily be able to find out a solution for those tricky questions. How can the movement of the water be stopped? By either substituting water of the beaker with 10% solution. By substituting water of the beaker with 10% solution, or substituting the solution of the funnel with water. Substituting solution of the funnel with water. Either of the two you can write. दोनों लिखना जरूरी नहीं है. You may write any one of the conditions. Still you will get full mark. And the last one, what should the components be substituted to make water salty? Muslin. तो यहाँ पे component substitute करके, now I say I want to change this. Or now I'm saying that how can, if in the previous condition I gave you that here it was 10% solution, here it was 10% salt solution. And I said that what can be the change to make this water a little bit salty? I want to make this water a little bit salty. What should I do? What should be the change in the experimental setup? मुझे ऐसा कुछ सोचना है where not only water but solutes also should move. Because by the way of the Parkman's membrane, water is moving if I change the concentration. Yes, we can change the concentration to make the water move this way. But मुझे water, I want the water to be salty. If I want the water to be salty, solutes also should move from here to the water. The solutes also should move. That means the salt molecules also should move, no, along with the water. तभी जाके this water will become salty. But instead of using the muslin, if I just use cellophane, I substitute it by cellophane. I don't use muslin, I use cellophane. तब तो वही पहले जैसे end osmosis नहीं होगा. और cellophane use करके I take this as 10% solution and I take this as water. Yes, the water will only run down. Is the salt running down? No, because I'm still using semi-permeable. I want this. What is the property of this membrane muslin? It is freely permeable. So it will allow not only the movement of water but also the movement of the salt molecule, solute. The salt will come slowly get dissolved in the water and the water will start tasting salty after some time. This water will start tasting salty. Are you clear with this set of questions, all of you? Clear with this set of questions, all of you? So after some time again, maybe in the next lecture again, I will give you some set of questions this way. By that time, please go through these concepts. See that you are able to apply the concept of endosmosis, exosmosis, concentration differences, and the concept of the different types of membranes that you have studied in all these different questions that you get. Today, let us start with another topic here, active transport. Let us start discussing this. What is active transport? What happens in this process? Active transport, almost the opposite concept that we are going to study today. Active transport. What was the definition of the word diffusion? We have studied definition of diffusion. What was it?
Higher concentration to lower concentration.
Movement from
Higher concentration to
diffusion. Movement from a region of higher concentration to a region of lower concentration, but of what?
Molecules.
Substances. वो कुछ भी हो सकता है, no? When we study diffusion, we always understand that we are talking about the movement of the molecules of a substance, and the substance can be gases, substance can be liquids, and substance can be solute also. Any three. Then what is the difference with osmosis you had learned? Only water molecules. ये याद रखना है. Only water molecules. But what is the similarity between diffusion and osmosis?
They have a higher concentration. They flow from higher concentration to low
Movement is in both the conditions. Movement is from a region of high to a region of low concentration. So that's the similarity. Now we are talking about active transport, a concept that if I now talk about the plant cells, we were already talking about the plant cells earlier. Now I talk about the plant cells again, and this plant cell, what do they contain inside? They have the sap. They have the sap. Now inside the sap, you already understand that there are so many salts and ions. There are so many salts and ions. So now whenever we are talking about all these different type of ions and salts and minerals are there, no? Manganese and zinc and potassium and sodium and all these and their respective ions are there, the radicals and the ions, all these which are dissolved. So the salts and ions which are dissolved in the cell sap, in any condition, this type of salts and ions are more in the the salts and ions whenever we are talking about. What was that one word we had used for the condition of the cell sap? The cell sap is, what did we understand? The cell sap is, there's one word I taught you the other day, hypertonic. Now what is the meaning of this we had learned? The cell sap is hypertonic. That means at any time, whenever I am comparing the soil water with the cell sap always has got a higher concentration of these ions and salts. Yes, there are enough amount of ions and salts here also. I talk about the presence of the zinc and the manganese and the magnesium, and all these are here also outside in the soil water. But if I check 1 ml of soil water, 1 ml of cell sap, this 1 ml of cell sap is always concentrated. When I compare, yes, this is more concentrated. This has got a lesser volume of water and more amount of solute ions now. I'm talking about this ions now. That's the reason we are saying that the cell sap is hypertonic. But this is a different concept altogether, where we understand that though the plant cells has got a higher concentration of ions inside the plant, inside their cell sap at any time, but the cells are still interested to take in more and more of these ions from the soil. It is something like us, more or less. No? Even if you have got a cupboard full of dresses, still we are buying. Still we are buying. No? It's happening for all of you. So the same is happening for the plant cells. The same is happening for the plant cell. The plant cell has got enough. A lot more than the surrounding water, but still it wants to grab enough. It still wants to take enough. It still wants to take enough. It still wants to take enough. So Are these ions now moving from a region of high concentration to a region of low concentration? No, because repeatedly I'm telling you that these ions are already in a lower concentration outside in the soil water and they are already in a higher concentration inside. So actually the movement should be from a region of low concentration to a region of high concentration in that case. But when the movement has to happen from a region of low concentration to high concentration, the movement will be difficult, no? The movement will be strenuous. Something like when you are climbing the steps, you are climbing a hill, it's difficult, no? But running down the steps is easy. Less amount of energy we need. Running down a slope is easy. When you are cycling also, when you are driving a car also, when you have to raise a hill, it's difficult. Even with the machines, it's difficult. When we are walking also, it's difficult. But running down is easy, no? Coming down the slope is easy. So same here. Moving from a region of low concentration to high concentration is difficult. Coming down from a region of high concentration to low concentration is easy, smooth. So here what happens, I said that the plant cell as it is has got a higher concentration of these ions and salts anytime, but still the plant cells are constantly trying to keep a reserve in them. The plant cells always try to keep those reserves in them by taking all the ions and salts from the surrounding medium. They are constantly trying to take them so that the plant cell never falls short of these ions. Why? Because these ions are necessary for the formation of the pigments. These ions are necessary for the formation of the enzymes and the proteins and the hormones that the plant should constantly keep preparing. A very simple example, the other day also I was telling you that why should the plants constantly take the minerals from the soil? The plants want to constantly take the minerals from the soil because the simplest thing to make you understand is the chlorophyll is a type of a pigment that the plant should have in abundance anytime. The plant should have chlorophyll and it's like chlorophyll, so many other different type of pigments and proteins and enzymes in surplus. Our body also has that, no, in surplus. So that's the reason, even though the concentration is high here, concentration is low here and high here, the plants are still interested to take them inside. But now, to take them inside, will this process be easy? No, the process will not be easy. But these plant cells also have got a specialized type of a organelle. What is this organelle known as? This is known as the mitochondria. And why suddenly I'm talking about the role of mitochondria is mitochondria is associated with the result with the production of energy molecule in the form of ATP. So now in this case, same like running up the steps, climbing an uphill. In any of the cases, we know the amount of energy that we also spend is much more. So same here, the plant cells also have to utilize a large amount of energy molecule. They have to spend a large amount of energy molecule to take these ions inside. And the plant always does that. The plant cells always does this. So now, if I have to talk about that, what is the definition? What is this particular process? This particular process where the ions and the salts solutes, in one word, solute, the process by which the solutes are moving from a region of low concentration to a region of high concentration by using energy molecule is known as active transport. What is active transport? Is it not the opposite of diffusion? Is it not the opposite of passive transport? The process of diffusion is also known as passive transport because the movement is happening smoothly, easy. The cells don't have to utilize any amount of energy there. The cells don't have to utilize any amount of energy there. It is just smoothly happening. Because if I now talk about the movement of the water, where is water more, in the soil or in the sand? Soil. So water is more in the soil. So what did we learn for the concept of osmosis? If it is water molecule straight away can enter into the sand. And this is what we will talk about as passive transport. What is the meaning of passive transport? Passive transport is a process where the movement of substances are happening from a region of higher concentration to lower concentration. Is there any expenditure of energy? Extra expenditure of energy is there? No. because high concentration say low concentration movement is as it is smooth. Solutes, especially all these ions, when they are moving from the soil water to the vacuole, they need energy. Let us write down the definition first. What is the definition of active transport? The movement of solutes. The movement of solutes, that is ions such as move from a region of low concentration to a region of high concentration using energy in the form of ATP. Using energy in the form of ATP. This definition is very important. Movement of solutes or ions. Movements of solutes, that is ions such as, any examples you can write. I have given you only 2 examples, sulfates and nitrates and chlorides and zinc and magnesium and manganese and potassium, all these, any example you can write. Just 2-3 example लिखना है, such as sulfates and nitrates and etc. Move from a region of low concentration to a region of high concentration. Move from a region of low concentration to a region of high concentration using energy in the form of ATP. Using energy in the form of ATP. Is this clear? Is this clear? What is active transport? Now for active transport, you don't have the experimental setups to understand, but there are many experimental setups to understand active transport also. We are not talking about the experimental setups much because that's not included in your syllabus, but the differences. Now, let us take one question. Try this. Write 2 differences between active transport and passive transport. That's a common question in your paper. Write 2 differences between active transport and passive transport. Make a column. Write down 2 neat points about active transport and passive transport. 2 differences between active transport and passive transport. Now, I'll give you the clue what to write. What are the points that you have to write? You frame your own answer. I'm going to ask you after 5 minutes what you've written. The first point you will write always is based on the movement, the directional movement. Second point you will write based on what is moving. What is moving? During the process of active transport, movement of what you are talking about. And during the process of passive transport, meaning osmosis, or it can be diffusion. Here for passive transport, you may talk about movement of, or movement by the way of diffusion. You may also talk about movement by the way of osmosis. So the first point you have to focus on when you write down the difference. The first point you have to focus on the direction of movement of the molecules. If it is active transport, from way to way, and if it is passive transport, then from way to way. So that's the first point. Second point, you will give examples that during active transport such and such molecules can move. What type of molecules can move during active transport? And during passive transport, what type of molecules can move? Chalo, right. Let me see what you all understand. Try to write very brief but very correct answers. Make a column and write down the difference. Finished? Done? I asked you to write two differences between active transport, passive transport. What did you write here? The first point.
Active transport is the movement in which the movement is from a region of low concentration to high concentration. Passive transport, the movement is from a region of high concentration to a lower concentration.
Very good. And what is the second point you will write?
Movement of solids such as magnesium and nitrates takes place
For
active transport, and for passive transport, movement of solids such as water takes place.
but khali water nahi. Passive transport can also happen for gases, because when you write passive transport, you understand the concept of diffusion and osmosis both. Passive transport is applicable for the concept of diffusion and osmosis both. Do you remember still what is the similarity? That's why I asked you this already earlier. What is the similarity between diffusion and osmosis? Movement is always from a region of higher to lower concentration, and molecules कौन-कौन से applicable है? If I talk about osmosis, water, and if I talk about diffusion, it can be solute, water, gases, anything. So when you write down the second point, you have to write active transport applicable for molecules such as iron, salts, minerals, etc. Passive transport applicable for minerals such as gases, applicable for molecules such as gases, solutes, and water. Are you clear about this concept? Active transport and certain few applications. And one more point. I asked you to write two. Of course, we discussed two. एक और point भी है जो
Energy diffusion.
energy. So 3 points you very much know. उसमें से any 2 points you write whenever asked or whatever is asked. So write down the third point also and be. For active transport, is there need of additional energy?
Yes.
Yes, there is a need of additional energy in the form of ATP. There is a need of additional energy in the form of ATP. For passive transport, no need. No need, or there is no expenditure of energy in the form of ATP. All of you are done? Now the next concept that we are going to study, turgidity and flaccidity. Remember, try to understand these terms, differences. Okay, these are fine differences. Turgidity and flaccidity, that is what we are going to talk about. What do you understand by turgid and flaccid? Let's take up. Turgidity and flaccidity are just two opposite conditions that the plant cells go through from time to time. So now, first of all, to understand a concept of turgidity. What is the meaning of the word turgidity? Now, say suppose this plant cell is in contact with the soil water. And again, we believe the similar condition. Again, we believe the normal similar condition again. We have been already talking about this. And I'm saying that yes, if this is a dilute medium, this is a dilute medium, and this is a concentrated medium. So what are we actually comparing? We are actually comparing the cell sap with the soil water. We are comparing cell sap with the soil water every time. And now we know that if the soil water is dilute and sap is concentrated, then there will be an easy movement of water in this direction. So the water molecule constantly moving in this direction. No? The water is constantly moving in. Will the water keep on moving for a long time? Yes, the water constantly keeps on moving. The water constantly keeps on moving. Do they ever come across or do they ever reach a stable state जहाँ पे normally water movement stop हो जाए? Generally नहीं. Why? Because this water is not staying here, no? This water is constantly getting distributed to the surrounding cells. ये जो water or the sap which is getting accumulated here in this cell, will it remain here? No, it's not remaining here. It is constantly getting distributed in all directions. So इसीलिए water is always less. Water is always more as long as the soil is well watered. हाँ, कभी-कभार situation ऐसा भी होगा when the soil is not well watered, when the amount of movement of the water may slow down if the soil is not well watered. But we are taking it as a normal condition that the soil has got adequate amount of water. Now what? Now the water is constantly moving, and as the amount of water is getting filled up, what will happen? Will the size of the vacuole remain the same? No, the vacuole is also going to expand. What was the membrane of the vacuole called? Do you remember? Tonoplast. Very good. Tonoplast. So ये जो tonoplast है एक single layer membrane है, and this is flexible to a great extent. So ये जो vacuole है, no, the vacuole has now extended to a greater extent. ये vacuole पहले यहाँ तक था. Now the vacuole is not till here. The vacuole has extended quite a lot. And this will go on and the vacuole will start extending. अभी इसके साथ, what are the other changes that is happening? The other changes that is happening is that this is what is this membrane? This is the cell membrane, no? And this is the cell one. So along with the extension and expansion of the vacuole, the cell membrane also starts moving towards the cell wall. Because now the cytoplasm is also, the volume of the cytoplasm is also increasing. It is not only that the volume of the contents in the sap or the volume of the contents in the vacuole increasing, certain amount of water is also getting flowed into the cytoplasm. So the volume of the cytoplasm is also increasing. So इसकी वजह से what will happen? This cell membrane will also start extending and moving and pressing towards the cell wall. So this cell membrane also will start pressing and moving towards the cell wall. But yes, as and when they are moving, now if I say that now the cell wall also has started extending and the cell wall is also now in the process of getting pushed towards the cell membrane, this is what is happening. Then yes, the volume of cytoplasm also increasing. Then what happens? A time comes when this process continuously happens. A time comes when the cell membrane is very close to the cell wall. The cell membrane has come very close to the cell wall on all directions and on all the four sides. The cell membrane has come very close to the cell wall, and the cell is not able to take in any more water. The cell is already having so much of water. The vacuole is fully distended with water. The vacuole is fully distended with water. The cytoplasm has already increased its volume so much. And what are the third change I told you? The cell membrane is getting pushed and pushed and pushed towards the cell wall on all the sides. The cell membrane is getting pushed towards the cell wall. The cell membrane is getting pushed towards the cell wall on all the sides, on all the sides. So at this time, the movement of the water slows down. Yes, at this time, the movement of the water slows down. And this is a condition when the plant cells are not able to take any more water. The plant cells are fully filled with water. There is no chance that the plant cells would take any more water. Something like, say, suppose when we eat a lot, quite a number of times it happens, no? We eat a lot or we have taken in so much of water. A time comes when we are not even able to take in a gulp of water or not even a single amount of food. Same that way, the plant cell at certain times it happens when they have taken in already so much of water that they are not able to accommodate any more water. So what is this condition known as, or what is the cell now known to be? The cell is in a state of the cell. What is the state of the cell? Remember these terms. What is the state of the cell? The cell is turgid. And what is this condition known as? The condition is now known as turgidity. Turgidity. What is meant by the turgid condition of the cell? Turgid condition of the cell is the state of the cell or the condition of the cell when the cell is not able to accumulate any more water. Why? Because the vacuole is already tight filled with water. Because the cell membrane is already pushing against the cell wall. The cytoplasm volume has already increased to its best. इससे ज़्यादा और नहीं हो सकता. But still, if the cell is in a very dilute medium where there may be even some more chances of entry of the water, then what may happen to the cell? The cell may start bursting out. The cell may start bursting out, if at all. Under this condition, yes, the cell may more or less manage, but after some time, still if the continuous movement of the water happens, the cell may burst. Does it normally happen? Did we ever see the bursting of the cells in case of the plants? Yes, quite a number of times we get to see that certain types of fruits and certain types of seeds, fruits, or quite a number of times in our own homes also we get to see, that if say suppose those raisins and all we have kept for a longer time in the water, yes, we get to see that their outer membrane have burst out. These types of fruits, especially the ones which are filled with water, grapes, strawberries, quite often they burst out. They burst out because as it is, they have got so much of water in them. So what did we learn? The first one, the first condition is what is meant by the turgid state of a cell. What are you supposed to understand? Turgid state of a cell which is already filled with water, a cell which cannot accommodate any more water, and this condition is called turgidity. This condition is called turgidity. Now, next is, so during this turgid condition, during this turgid state, what are the features? The cell membrane is pushed against the cell wall. The cell membrane is pushed against the cell wall. And it is also understood that when the cell is in a turgid state, it is more or less balanced. इसीलिए movement of water slow down हो जाता है. When the cell is in a turgid state, the amount of water here, the amount of water here is almost balanced, not fully, but almost balanced. इसीलिए movement of water slows down. So you get a statement sometimes in the paper. During a turgid state, the cell is balanced. Yes, it's a true statement. During a state of turgidity, the cell is in a balanced state. Is this statement true or it is false? It is true. इसीलिए, when it is balanced, in the previous experimental based question that I gave you, how can you make the movement of the water stop? Only when on either side of the semi-permeable membrane there is a balanced state achieved. तो यहाँ पे भी fully balanced state achieved नहीं है, but why does the movement stop or why does the movement slow down? Because on either side of the cell membrane, more or less a balanced state is achieved. During a turgid condition, the cell is known to be more or less in the balanced state. So this is the first thing we understand, that during turgidity, what happens. Now next is, when we talk about the condition of turgidity, we also side by side try to understand that, yes, I said that if this turgid cell is still in a dilute medium, then there is a possibility that the cell may burst. Then there's a possibility that the cell may burst. But now, what do? Is we now keep this cell or put this cell in a different medium. Where are we putting this cell? We are now putting this cell in a medium not like this. If these type of cells are now kept in a or put in a medium which is little bit concentrated. Now, how can we make the soil water concentrated? By adding some nutrients or minerals there. If I add some fertilizer, I add some minerals there, then the soil water is becoming concentrated. So now, in that case, if I talk about the same root cell, say suppose now I am saying adding some minerals to the soil water, adding some minerals to the soil water. Then what am I trying to make? I am trying to make the outer environment of the plant cell something concentrated. And when now the outer medium is concentrated, what will happen? The movement of the water will start happening this way. The movement of the water will start happening this way. समझ में आ रहा है? So first of all, we understood that the water movement is inwards, and that is what is responsible for the turgid state of the cell. During our turgid state of the cell, I gave you three conditions to remember. What happens to the vacuole, what happens to the cytoplasm, what happens to the cell membrane. How can we reverse the turgid state of a cell? Turgid state को reverse कैसे कर सकते हैं? By making the outer environment of the cell concentrated. So if I'm talking about the real situation, I have to just add some minerals or some fertilizers in the soil, and then now the water in the soil becomes concentrated. Is it not that the direction of the water movement will change after some time? Yes. Now the plant cells, which are having lesser amount of minerals and ions here and more here, say suppose I talk about one single mineral or ion, the amount of nitrates, if it is this much in the sap, now the amount, because I have already added so much of fertilizers or minerals, now it is more here, no? And since it is more here and water is so much here and water is less here, the direction of the water movement will be this way, from the cell to the soil. What is this type of movement of water known as? When the cell is giving away water, what is this type of osmosis known as? Exosmosis. So just after this, after bringing about this change, what do I see? I see exosmosis happen. And as a result of exosmosis, now what? As a result of exosmosis, what will happen? The cytoplasm will, or the first of all, the vacuole. Let us talk about the vacuole. First of all, the vacuole will start shrinking. First of all, the vacuole will start shrinking. What is the other change that we can see here? The cytoplasm also is losing water. No? The cytoplasm also will lose water. If the cytoplasm is losing water, what is the next change that we should bring into our notice? The next change that we should bring into our notice is that the cell membrane will also start getting shrunken. Pehle cell membrane was almost touching the cell wall, pushing against the cell wall. Now it is getting separated from the cell wall. And so much of water lost, slowly, what will happen? The cytoplasm shrinking, the vacuole shrinking, and the cell membrane is getting separated from the cell wall. So when this condition is happening, this state of the cell is known as flaccid, and the condition is known as flaccidity. The condition is known as flaccidity, just the reverse of turgidity. How do you define the flaccid condition of a cell? What is flaccid condition of a cell? the cell which has given away large amount of water, and as a result, the vacuole, the cytoplasm shrinks, the vacuole and cytoplasm both will shrink, and the cell wall or the cell membrane separates from the cell wall. The cell membrane is separating from the cell wall. Okay? So now, if I say, now this is the real situation I'm saying. If in the soil itself, जो plant cells में normally we have say turgidity happening, तो flaccidity हम कैसे ला सकते हैं? By adding these minerals. But if I talk about an experimental situation, this type of questions we will get again. Experimental situation hai. I talk about a beaker again. I talk about a beaker, and this beaker has got water, and these are some plant cells that I was taking for an experiment. So initially I had seen that these plant cells, by absorbing water, they have become turgid. Initially, that was the first part that I explained to you, concept of turgidity. Initially, water was entering and the cell was becoming turgid. So यहाँ पे water था, and यहाँ पे तो normally sat. Now, in this experimental setup, you want to make the outer environment of the cell concentrated. यहाँ पे कैसे करेंगे? Water को substitute करना है. Okay. पहले हमने water लिया था, अभी water को substitute करके what can we add to make the outer environment of the cell concentrated? कुछ भी no, you already know now so many examples. क्या add करेंगे? What can we put here? Give me anyone idea. What can we put here to make the outer? मुझे outer environment of the cell को concentrate करना है because I want to make this turgid cell flaccid. मुझे turgidity को reverse करना है. So यहाँ पे तो normal situation में मैं तो I had added some fertilizers, I had added some minerals. यहाँ पे experimental situation में मैं क्या करूँ? Salt. By adding some salt, very good. By adding some salt, small amount of salt you add, or small amount of sugar you add, anything. तो ये जो water हमने लिया था, यहाँ पे small amount of salt add कर दो. Small amount of sugar add कर दो, 1 teaspoon of sugar, 1 teaspoon of salt add कर दो, then the outer environment of the cell is becoming concentrated. Will the turgid condition now start getting changed to the flaccid condition? Now this cell will start not taking water, rather the cell will now start giving away water, and this vacuole will not remain like this, the vacuole will shrink rather. Is this concept clear? So what did we understand so far is, when is a cell turgid, when is a cell flaccid? Normally, naturally also the plant cells are turgid. And when the distribution of the water minerals takes place, yes, they again revert back to that flaccid condition again. Now, say suppose when we are talking about nowadays it's very evident to see this concept of turgidity and flaccidity, especially in the smaller plants. In the smaller plants, as I said, if you talk about the plants that you have in your own windowsill also, the small herbs, some of us we have got those tulsi, and some of us we have got those money plants, and some of us we have got this different type of small flowering plants. No? In this small herbs and shrubs, nowadays it's very evident because afternoon is very heated up. And the evening again is quite cool, the morning is so cool. No, in the morning you will see that the leaves are so fresh because the cells are turgid. But afternoon you get to see that the leaves are so dried out and wrinkled and drooping. Why? Because the cells are flaccid. So flaccid and turgid are just opposite of each other. So what are the three conditions? Again I repeat, remember again, during a turgid state of the cell wall is distended. The vacuole is distended, the cytoplasm has increased its volume, and the cell membrane pushes against the cell wall. And इसके just reverse होता है when you talk about flaccid. So these are the two terms. Now something more that we try understanding. That this is the cell again. We are talking about the cell, a plant cell again. And in this plant cell, just now we were saying that in this plant cell, there is the cell wall, and the vacuole. And now I try to understand that yes, if it is the turgid state, तो turgid state में क्या हो रहा था? Adequate amount of water was entering. Adequate amount of water was entering. Yes, the vacuole was becoming bigger. The vacuole was becoming bigger. And inside the sap, there is adequate amount of water, there is adequate amount of salts, there is adequate amount of minerals, there is adequate amount of ions. The volume of this is also increasing. जैसे कि water is constantly entering, the volume of the vacuole, first step increasing, cytoplasm increasing, cell membrane. So already we have come across this. Now, yes, again I see that due to the continuous inlet of the water, due to the continuous movement of the water, what is also happening is that this cell membrane is constantly moving towards the cell wall, and due to this, as the cell membrane is constantly moving towards the cell wall, there is a stable state achieved, and the cell has a great greater amount of flexibility, or the cell can hold that water for a long period of time. I repeat, I said that when the continuous movement of the water takes place, is it that the cell is turgid and then after some time it bursts? No, the bursting of the cell is the last possibility that takes place, where the cell is not able to manage. But what is the cell trying to manage? How does the cell try to manage? First of all, when the cell contents start increasing, there is a pressure which has been exerted by the cell contents on the walls of the cell. A pressure is getting exerted by the cell contents, because these cell contains, the contents of the cell is increasing, no, in volume. The cytoplasm is increasing volume. So right now I'm only talking about the contents of the vacuole, but you have to understand is the cytoplasm also. So now I say that ये जो vacuole है ये बड़ा होता जा रहा है क्योंकि salt and ions इसमें increase होता जा रहा है, and this volume of the cell is exerting a pressure on the membranes and the wall of the cell. So we just put it this way, that the cell contents exerts a pressure on the cell wall. Very simply we say that the cell contents exerts a pressure on the cell wall. तो अगर ये pressure continuous होगा after a particular period of time, it is evident, no, that the cell wall will break. Yes, the membranes will start breaking. It is evident. अगर ये pressure continuous चलता रहा, then yes, the membranes of the cell will rupture. But again I said, इतना easily rupture नहीं होता है. क्यों? Because as and when this pressure starts exerting, there is a reverse pressure which starts exerting from the wall towards the cell contents. It is something like a tug of war. You also pull and I am also pulling. So that way, this contents are exerting a pressure on the wall, and the wall is also exerting a pressure on the contents. What is the state of the cell? Turgid. The state of the cell is turgid. क्या ये flaccid condition में कभी होगा? नहीं, flaccid condition में तो ये सारे pressures exert ही नहीं कर पाएंगे, है ना? So I am talking about a turgid condition of the cell when the volume is full in the cell, vacuole is filled in the cell. This is exerting a pressure on the cell wall, but counter, a cell wall is also acting a pressure on the cell contents, and this is what is responsible for the stability of the cell. I have been repeatedly telling you, turgid state maybe the cell does not burst so easily. Q, what is the reason? What is the reason for the balanced state of the cell for a long, long time? Especially if I'm talking about the aquatic plants. See, terrestrial plants, it's easy for them to maintain this stress. But what about the aquatic plants? The lotus, all these different type of hydrilla, etc., etc. Aquatic plants which are already there in the dilute medium. Do we get to see that the cells of the aquatic plants are always dead and always rupturing and always breaking, bursting? No, they don't. And how do they exert or how do they tolerate that so much of pressure? Because of these two pressures. What are these pressures, first of all? The first, the pressure, the force which is exerted by the cell contents on the cell wall. This is called That is called
Turgor.
Turgor pressure. Turgor pressure. Example, I'm sorry, the conditions of Turgor pressure we are going to learn in certain other experimental setup. Pressure of the cell contents on the cell wall, Turgor pressure. What is the reverse? What is the counter? Pressure of the cell wall on the cell contents, and that is what is known as wall pressure. So when we are talking about these two pressures are counteracting. Both of them are acting together. One from the wall to the cell contents and the other from the cell contents to the wall. And this is what is constantly happening in the plant cells. This is what is responsible for the balanced state of a cell. Right, I'll take one question here. See that you are understanding, see that you are following all the terms. Right, one question. How does a cell of aquatic plants, how does a cell of aquatic plants, why do you write with such a light coloring? Huh? Don't write with this type of colors, no? Write with a dark ink. Use a pencil which is dark. Use a pen which is dark. Huh. What I said, read out what I said.
How does this?
How does a cell of the aquatic plants maintain their stable state? Maintain their stable state. Maintain their stable state. Answer. Cells of aquatic plants are always in contact with a hypotonic medium. Full stop. Due to full stop. Next sentence. Due to continuous endosmosis. The volume of the vacuole and cytoplasm. Increases, which exerts a pressure on the cell wall. Hyphen, known as Turgor pressure. Full stop. In turn, T-U-R-N, in turn, the cell wall exerts a pressure on the cell contents. Hyphen, known as wall pressure. Full stop. These two counteracting forces, keep the cell in a balanced state. Okay, is this understood? So for 2 marks, how does a cell achieve a balanced state? Especially now, we are talking about cells of the aquatic plants, remember. Again, I repeat, the terrestrial plants don't suffer from this type of a problem much because the soil sometimes is dry also. No? The soil will not have abundance of water every time. The terrestrial plants don't have this problem much because at times the water is more in the soil, at times the water is less. But this problem is majorly with the aquatic plants because they are always in a medium where there is surplus of water. Itna sara dilute medium, itna sara water, and the water is constantly moving in. So how does the cells of the aquatic plants resist themselves? So under normal condition, the cells of the aquatic plants should be always breaking and rupturing. No, that does not happen. So how do they resist themselves? So this is what you should always write. That these two pressures, the turbo pressure and the wall pressure, always counteracting each other, and that's how they are always maintained in the balance state. So now what did we learn? That when we talk about a plant cell, how can the cell achieve the turgid state? How can the cell achieve its flaccid state? Relating to that, now we also talk about another condition, and as now I'm saying that this is a cell and this is the cell membrane, cell wall, and the vacuole. And now you tell me, what should be the condition of the surrounding soil? Now this is the soil, and this is my plant cell. What should be the condition of the surrounding medium of the cell? What should be the condition here in the soil so that there is adequate amount of exoosmosis happening? Higher concentration. No, that means when we are, say, suppose we talk about a condition of in the real condition, a plant cell. Generally, they don't give away water. Plant cells normally they take water. Do they give away water? No, they don't give away water. But under normal condition, how can we set up or how can we bring about the change when the root cells are giving away water to some extent? Yes, the root cells may give away water to some extent when the soil is supplied with large amount of fertilizers. When the soil has been supplied or sprayed with large amount of fertilizers, yes, at that time, the soil water is becoming so much concentrated. Will the cell give away water? Yes, the cell will give away water at that time. Yes, the root cells will give away water at that time. So this is the real situation that I'm talking about. But now, let us just talk about experimental setup also. This is a plant cell. And here we are talking about the water outside. We are talking about the water. Normally, the water will move in. Normally, the water will move in. Same a bigger experiment or anything. So in an experimental setup, I'm taking water outside and the plant cells. Normally, the water movement is this way. If this continues, we suffer or we understand the cell enters a turgid state. Now, I said this water, I add enough amount of salts. I add enough amount of salts, then I get to see that the movement of the water is this way. And slowly, the water movement is continuous. Slowly, the water movement is continuous. And what will be the first change that we get to see? The vacuole will not be this way. The vacuole will start shrinking. What is the next change? The amount of the cytoplasm. Will it remain this way? No, the amount of the cytoplasm also is not remaining this way. If this process continues, the amount of the cytoplasm will also become lesser and the cell membrane is far away from the cell wall. The cell membrane is far away from the cell wall. So now, what are the two conditions I told you? The two conditions would be number one, vacuole shrunken. Cell membrane separated from the cell wall and cytoplasm also shrunken. So when all these features we talk about, say suppose I'm talking about the first condition is what? The vacuole. We talk about the vacuole reduced in its volume. The vacuole is shrunken. The vacuole reduced in its volume. Next is the cytoplasm reduced in volume. And what else? Very prominent to remember is the cell membrane separated from the cell wall. Already you have talked about this. The cell membrane separated from cell wall. This actually we have learned with reference to flaccidity, but now we are saying when all these conditions happen for a longer time, when the cell wall is also affected, meaning the process of exosmosis is happening for a longer time, when the cell wall is also affected, the cell wall is also shrunken. The cell wall also, generally the cell wall is a rigid layer. The cell wall is a rigid layer which easily does not get affected. But now the exosmosis is so big, so continuous, happening for a long time. Then what? All these changes happen one after the other. This condition of the cell will be termed as plasmolysis, and the cell is known to be plasmolized. Then what is the difference between flaccid and plasmolysis? Is there a difference? There's a big difference. Flaccid condition is just the one when the cell has just started losing some water. The vacuole may become a little bit smaller. The cytoplasm may shrink down a little bit. But when the shrinkage and the movement of the cell membrane from the cell wall is big enough, and the cell wall also has started shrinking little bit, that is a condition. Meaning it is far away from flaccidity. Far away from flaccidity. Flaccidity may agar exosmosis is just 30%, plasmolysis may exosmosis is almost about 50%, 60%, something that way, if I try to make you understand. So flaccidity, exosmosis kam hota hai. Plasmolysis में exosmosis content of or the extent of exosmosis is even more. Plasmolysis. What is the basic definition of plasmolysis? A condition of the cell when the cytoplasm and vacuole has reduced its volume and the cell wall is shrunken. Cell wall का effect होता है. The cell wall is shrunken. I'll give you the same 15 minutes break. Not to worry. I'll just complete with this. अभी ये हो गया plasmolysis. But मुझे अभी ये cell को, this is my experimental cell. So what I took some cells in a medium which has got water and some salt. 10%, 5%. Let me write. Water and I have taken some 5% salts. Small amount of salts I took. Abhi, this is plasmolized. Plasmolysis ho raha hai. So you have to understand, plasmolysis mein the vacuole is shrunken, the cytoplasm is shrunken, cell wall, cell membrane, cell wall is separated hai, and cell wall also has started shrinking. Abhi, before the cell dies out, abhi ye agar काफी देर तक चलेगा, the cell will die. But before the cell dies out, I want to revive the cell. I want to retrieve back the condition. So मैं क्या करूँ? Should I add even more cells or salts, or should I add little bit of water? Little bit of water, or rather try to put the cell in a medium where there is enough amount of water. Minimum salts होना चाहिए. मतलब keep adding water so that the medium is now dilute so that adequate amount of water movement now start taking place. So ये process जब शुरू हो जाएगा, the cell membrane will go back and start placing itself near the cell wall. So सबसे important क्या हो जाएगा? Now the cell membrane will start taking up position something this way. What will be the next? The cytoplasm will increase in its volume. Yes, the cytoplasm is increasing in its volume. This is the second change. And what is the third change? The vacuole is becoming bigger. So this is the third change. When the cell is again retrieved, when the cell is again going back to its normal situation, what do we talk about this as? This is now known as deplasmolized. The cell is deplasmolized. Process is deplasmolysis. Plasmolized is associated with what type of osmosis? Exosmosis. Deplasmolysis is associated with what type of osmosis? Endoosmosis. Okay? We'll try to understand after the break what are some of the applications of plasmolysis, deplasmolysis. Okay? Take a break. We'll continue again after the break.
मतलब मैंने सिर्फ 3 प्रोटीन को भाई फ्री टिकट इसके पापा की तो हमारा फ्री टिकट नहीं लगा ना बेटे
अजय लेवल देखा
अच्छा उसका 12 फ्री टिकट है उसका मेरा दोस्त है वो है
जिसको
अबे मार्वल कौन देख रहा है ना मैं जान तू गया मैं जाके आ गया उसको लग रहा है भाई ये भी नहीं चल रहा है चलिए क्लास में खेलना है
10 का तरफ लेके
तो मैंने पूरा ना वापस डाउनलोड किया
दस मिनट बात करते हैं।
10 मिनट
हर वीक टिकट के लिए तो
किसी 4 मिनट का क्वेश्चन होता है 4 मिनट लास्ट में ब्रेक के टाइम बस 4 मैं 15 मिनट प्रेजेंटेशन में अरे अभी मैं बाद में पर फिर वो एक आईडी कार्ड से वॉशरूम करना पड़ता है वो उनके पास है ना